So as in the last video, we were done with the exceptions of first law of Mendel, which was the law of complete dominance. So today we are starting our second law of Mendel, which is the law of segregation. As you can see here, topic, law of segregation, which is the second law of Mendel, right? So what he did for the experiment, right? He cross-pollinated the selected pea plant by removing the anther. Like, for suppose if this is a pea plant, wait, let me write here. For suppose this is not a pea plant at all, but for suppose think that this is a pea plant. This is a pea plant, and what he did for that, he removed these anthers. Like, as you can see here, they are cutting these anthers from this, right? So by removing the anthers from one flower to prevent self-pollination, like we know that flowers are the bisexual, right? They have ovary and they have pollen grains or anthers with them too. So for preventing self-pollination, what he did, he removed anther from this flower, as you can see here. So, and dusting pollen grains from anther plant onto the flower. After doing this, like after removing this, as you can see here in this picture, this is more clear, right? They are removing anthers first. These are the anthers. Wait, let me use another color of the marker. So first of all, what they are doing, they are removing anther. This process is called what? This process is called emasculation, right? So removal of anthers is called emasculation. Wait, let me mark here. As you can see here, this is emasculation, right? So they have removed these anthers. This process is called emasculation. Now they have to prevent that too, like pollen grains will be attached to this. So for preventing the pollen grains to be attached with this, what they did bagging. Wait, let me make it this. They bagged it. They bagged this flower. So this process is called bagging. Wait, let me write here. This is called bagging, right? They bagged it. But now what they did, they took another flower. Now this is reacting like a female, right? Because anthers are removed from this. Wait, let me write with another color. Yes. Now this is behaving as female because anthers are removed. Now it has ovary, right? So now what they are doing, they are making cross with of this plant with this plant, so that pollen grains can go to this plant and can fertilize it. As you can see here, transfer of pollen, which is the pollination. This process is known as pollination. The transfer of pollen from anther to the stigma, right? So this is cross pollination because we are having this another flower and this is another flower. So this is cross pollination, right? So this process is known as dusting, while we are taking these pollen grains and dusting them onto another flower. So this is known as dusting, right? So now we can say that now these will be fertilized, right? So he did this process. So next stage was to cross pollinate plants with different characteristics. Like initially what he did, initially he cross fertilized plants with the same characteristics. Like if I have one plant with a tall height, and other plant I do have with the dwarf height, right? So this is the one characteristic only. What is the characteristic here? Characteristic is here the height. Like either it can be tall or dwarf. So initially he cross fertilized plants with the same characteristics. Like if this is a tall, this is a dwarf, the characteristic is the same. Like the character is the same to ensure that the plants were true breeding. If they will be making cross between them, the result which will be formed of these plants will be the true breeding. So let me make down what he did, initially cross fertilized plant with the same characteristic. Wait, I'm writing here. Same characteristic. Or you can say that they were having only one trait. Right? One trait. Now let me make here. If this is a plant with tall height and this is a plant with tall height too, because we are talking about the same characteristics here or only one character, right? When we make cross between them, what we'll get in the result, that would be tall, right? The plant which will be produced will be the tall. So what do we say this? This will be called as true breeding. Right? Wait, let me mark here. Okay. So the next stage was to cross-pollinate plants with different characteristics. Now we are going with the different characteristics. Wait, let me write here. So, for suppose this is a plant of tall and here we have dwarf, which we represent with the small t, right? So these are both different characteristics, such as one with the tall height and we cross to one with the dwarf height, right? So these initial plants, like when we will making cross, not cross, but these are what here? These are the parent one. These are the parental generation. This is parental generation. Right. So resulting of these, the offspring. Wait, let me mark here. Resulting of this will be what? Will be T and T. Right. So offspring will be the also tall because of this T will be dominant over this T. So now we have to self-fertilize this. Now we have to self-fertilize, right? Means the same plant will be doing self-fertilization. Means the same T and T. Now this is what? This is we have filial 1 generation, or we can simply write here F1 generation, right? So let me mark here. This is filial 1 generation, right? Now what will happen, right? So now there will be self-fertilization or self-pollination to produce F2 generation. This self-fertilization continued until the traits were established for many generations, right? In the case of this tallness and dwarfness, the Mendel P experiment showed that a cross between a tall plant and a dwarf plant produce only tall plant. Why? Why only tall plant? Like then he was so confused. Like he was saying, if I'm making cross between the tall plant and the dwarf plant, I am just getting the plant of tall. Why not short? Because these both are tall. Because this capital T is dominant over this small t, and this capital T is also dominant over this small t. However, the F2 generation threw up a surprising result. The tall plant, or not the tall plant, but we are going with the dwarf plant variant appeared in a quarter of this generation. So right now what he did, let me tell you. Okay, I'm using some marker. Alright. So from here now the separation of this gametes happened. Like now this will be separated. Separation of gametes will be happen first. So one gamete, two gametes they'll be forming. Wait. One capital T and other gamete it will be producing, that will be small t. Right. Again, same with this case. One gamete it will be producing, it will be capital T, and another gamete it will be producing will be small t again. Right. Small t. So these are the gametes, right? Wait, let me mark here. This is what? These are the gametes. These are the gametes. Because first step is to separate the gametes so that it can produce the offsprings. Right. Now what will happen? Let's see. Like the F1 generation, which is this, you can see here, all had the capital T and small t combination, right? And so were all tall ports variants. Also, importantly, all had a 50% chance of passing on the recessive this small t allele to the next generation, right? So the best way to express what happens in the F2 generation is with a type of diagram which is known as the Punnett square, right? Here I am making the Punnett square. Punnett square. Right. So now let's see what is Punnett square basically. As the Punnett square shows offspring with a dominant T, which is here. The dominant T gene will always have the tall ports with only the offspring containing the small tt combination having dwarf plants, right? So this is why Mendel only found the 1/4 of the F2 generation had only this dwarf height. However, all but 1/4 had at least one this small t allele, and so this would be continued to be the passed down through the generation, right? So I am making here the Punnett square, as you can see here. Let me make the line. One line. This is another line. And this is another line. Here I have made the columns. Now I'm just going to make the rows. Okay. Here I am making rows. I need to make 3 rows, right? One. This is the second one. And this is the third one. Right. So here I have made the rows too. Now for finding out how many we have found the tall plants and how many we found the dwarf plants in the F2 generation, right? So I'm just going to find it out by the help of this Punnett square. Wait, let me mark here. By the help of this Punnett square, I'm going to find it. So for suppose if consider this as a male, consider this as a male, and consider this as a female. Right. Because this F1 generation will be also known as the parental 2 generation, right? So now I'm just going to write it here. For suppose this is the side of male. Here I have to put the gametes of male, and here I have to put the gametes of female, right? Female. Wait, let me write here. Female. So now the capital T and small t. I have written there in the separate columns. So now I'm just going to write this T key in the rows. So this capital T and this small t, right? So now I'm just going to change the color so that I can write the office springs. I'm going to write this with this. Okay. So TT. When the TT will be crossing, they will produce all tall, because tall will always produce the tall. If I'm making the cross of this capital T and small t, it is also, we know that this capital T will be dominant over this small t, so this would also be tall. This small t and this capital T would be capital T and small t, which we know that this will be also tall because this capital T will be dominant over this small t, right? So this small t and this small t will be small t, right? And we know that this would be dot because these both are the pure breeds or the two breeds, as you can see here, right? Because they are having only one character, not only one trait, which is the darkness, right? So as you can see here, now let me there with this color. Okay. Now what they'll produce? They'll produce 4 different offsprings. One is of TT. So let me write here capital T and capital T. Another is capital T and small t. So I'm making here capital T and small t. Another we do have, which is also capital T and small t. So making here capital T and small t. And last we have, that is small t and small t. I'm just going to make here the small t with the small t, right? So I have made here what we got here, like this. Now I'm just making the top, right? I should go with the yellow color so that, okay, this one t, for suppose, has came from here, and other this t, capital T, has came from here, right? Now this capital T, for suppose it has came from here, and this small t has came from here, right? Now this capital T has came from here, and this small t has came from here, right? Now this t has gone there as well, and this t has gone with this plant as well. As you can see here, now this generation will be known as what? This will be known as F2 generation. F2 generation, right? This is the F2 generation we do have. Right. Let me mark here so that you will know 2 generation. So now I'm just going to find out the phenotype ratio and the genotype ratio of this, right? So let me see. Okay, now I'm going to find out the ratio of phenotype and genotype with the help of a little square, right? Okay, let me use color, a different color now I'll be using. Okay. So now here we have, now first of all we are going with the phenotype. So let me write here phenotypic ratio. Okay, let me mark here. We are going with the phenotype ratio first. So what we have the phenotype ratio, right? So this would be, we know that capital T will be tall. So we do have one tall. Capital T and small t will be also tall, because we know that this capital T will be dominant over the small t, right? Same will be happening with this case as well. So here we have another tall plant. Okay, so how many tall plants we have gotten? 1, 2 and 3. So 3 we do have tall plants. Let me write here. This 3 is the ratio of what? This 3 is the ratio of tall plants. 3 is the tall plants ratio. Now I'm just going to find out the dwarf plant ratio. This is the tall plant ratio. Now we are just going to see the dwarf plant. How many dwarf plants we have gotten? As you can see here, only one. Small t will produce what? Will produce the dwarf plant. So here I can write 3, which is for capital or for tall, and 1, which is for what? Which is for dwarf. So as you can see here, the phenotype ratio, 3 will be tall in the F2 generation and 1 will be dwarf. So this would be tall, this would be tall, this would be tall, and this last will be dwarf. So here we just got that when we self-pollinated these both, this one and this one, what we got 3 tall plants and we got only 1 dwarf plant. So now I'm just going to find out the ratio of genotype. Let me find here. So now I'm just going to write. So genotypic ratio means the same genes and the different genes. So here you can see this is capital T, capital T. Now we don't have capital T and capital A in any box, right? This is only one. So capital T and capital T. This we do have only one ratio using another marker. Now this capital T and this small t, and this capital T and this small t, which means we do have 2 capital T's and 2 small t's, right? Capital T and small t, we do have 2, right? As you can see here, one this, another this, right? So just going to change the color of marker. Yes, as you can see here, we have 2 small t's too. So here I'm just going to write here the ratio of 2 small t's is also 1, right? So what we got, the genotypic ratio of this is 1 ratio 2 ratio 1. 1 is to 2 is to 1. 1 is for tall, 2 are also tall, but the genotypic ratio is changed. As you can see, capital T and small t will also produce tall, and small t will be dwarf, right? So basically this was the second law of Mendel, right? So in this, first of all, the separation of gametes happened, then the offsprings were produced, right? So this was all about the second law of Mendel, which was the law of segregation.